PhyloLM is a new way to compare and understand how different large language models are related to each other. PhyloLM uses techniques from biology to measure how similar or different their outputs are. This creates a kind of family tree or dendrogram. In this video, we are going to install this PhyloLM locally, and then I'm also going to how it works in a Jupyter notebook. We will also check out the relationship and how different models are related to each other. Philo, by the way, comes from a Greek word phylogenia. It means origin or evolution. In biology, phylogenetics is a study of the evolution and history of relationships between organisms. In the context of large language models, PhyloLM applies this concept to study the evolution and relationships between language models. So coming back to this PhyloLM, this creates, as I was saying, a kind of a family tree or a dendrogram, as you can see in this diagram, showing how the models are connected. This method can also predict how well each model will perform on certain tasks without needing to know all the details how they were trained. This can save time and resources when evaluating LLMs. Think of it like a tool to map the evolution of language models, helping us understand how they are connected and how capable they are, even if we don't have all the information about how they were created. Imagine you are studying how similar or different humans are based on their genes. First, you choose specific genes to focus on, like eye color or height, and then you look at the different versions of these genes. And these different versions are called as LLS in each person. Compare the LLS between people to see how similar or different they are. Then use a specific formula, in this case night distance, to calculate how far apart they are. And then use that information to create a family tree or dendrogram showing how related they are. When we apply it to large language models, as you can see on the diagram, first we select specific genes like model architecture or training data. Then we look at the different LLS, which is version of these genes in each LLM. And then we compare the LLS between LLMs to see how similar or different they are. Then use the same formula to calculate how far apart they are, and then create a family tree or dendrogram showing how related the LLMs are. In simple words, at very high level, LLS are different versions of a gene, just like how you might have a different versions of the eye color gene than someone else. LLMs can have different versions of their genes, like model architecture or training data, and this analogy helps us understand how to compare and study LLMs in a way similar to how we study human genetics. Also, there is a very finely written paper which I will drop the link in video description, which describe it in great detail, so do read it out. Now let me take you to my terminal in GUI where we will be installing this, and then we'll see how it works. So this is my Ubuntu system, and for this one I'm using NVIDIA RTX A6000 with 48 GB of VRAM. Both of these are courtesy to Massed Compute, who are sponsoring them. If you're looking to rent a GPU on affordable prices, I will drop the link to them and also a coupon code of 50% discount on a range of GPUs, so please do check them out. Okay, so that's said and done. Let me clear the screen here, and I will create a Jupyter notebook in my Conda environment first. I'm just calling it Philo. So let's wait for it to get created, and that is going to keep everything separate from my local system. Okay, so Conda is installed. Let's also install the Jupyter notebook here. Let's wait for it. And that is done. Let me clear the screen, and now let me launch the Jupyter notebook. And that is going to start the notebook in my browser. There you go. Let's wait for it to load. And there we have. Let's open a new notebook. First up, let's install some of the prerequisites, which are usual suspects like torch, transformers, and all that stuff. I'm also including the BioPython for this. And BioPython is primarily a freely available Python tools for computational molecular biology and bioinformatics. You can use it for computational aspects of molecular biology. You can also scan the GenBank database for specific sequences. Plus, you can also parse PDB files. You can do alignment and other evolutionary comparisons between different biological sequences. Plus, you can also perform phylogenetic analysis on various biological sequences with the help of this BioPython library. Amazing library, by the way. I might do another video on it separately, though it is not um, strictly related to AI, but it's a very, very interesting library. Okay, so let's wait for all of these things to get installed. They will take a bit of a time. Most of them are heavy libraries. And don't worry about the code. I'm going to give the link of this notebook in video's description. All the prerequisites are done. Next up, let's import the stuff which we have just installed. And next up, let's specify some of the models where will be testing it. And you see that we have got some Pythia related and then tiny stories related small models just to make sure that we have representative data. And then we have defined our genes and probes. Okay, so let's run it. It is just going to set the variables, by the way, nothing else will be downloaded shortly. And now let's define some of the functions which they have defined in their paper, in their repo. Don't worry about that code that much, because all it is doing is it is just looping through some of the phylolm through the gene set and all that vectors and forms. So just grabbing the model and tokenizer as we have defined above in the loop. TQDem is showing the progress, and for the phylolm, as I said, it is just generating different versions of the genes, LLS. And then this is all from their paper where it is drawing it on few axes, doing some Python magic again, nested loops here, and then checking that the similarity function which I described earlier, and then it is computing it all and then giving it the similarity matrix. So let me run it quickly, and that is all done. Now if you go through the paper, they have defined some of the benchmarks to construct the genome, and we'll be using this command to generate the genome. And it is slightly different method than the paper, letting the model generate the genome themselves by sampling a few tokens from an empty context. One model is sampled uniformly among the set of selected models to generate a new gene, and this process is then iterated g times to generate the full genome. So let me run it, and you will see that it is going to download the models, their tokenizers, and all that stuff. It is going to take a bit of a time depending upon the GPU you are using, because we have specified a lot of model, though they are very small, which is great. And it is also going to show you the progress, and hopefully they will fit it on my GPU. They are small, but there are a lot of them. So let's wait for them to finish. And we can click on this show more outputs, but I'll just let it run. All the models are downloaded, which is great. And now let's do some of the genome visualization. Let me run this loop here, and you see, there you go. So it is just using the models on some of the sample dataset output, telling all the genes and then different models, and we can just go through to see how these genes are related to each other, and then it has built upon the possible story. Also, it is giving us some of the keywords here. Very fine. Okay, so let's also now get the sim metrics and stuff. And this part where it is compute all the genes and everything, this takes long time. So especially if you are using CPU or a T4 GPU in Google Colab. But by the way, you can try it out on Google Colab if you like, on free T4 GPU, but it is going to take long time. So let's wait for it to finish, and then we will proceed further to try to plot it. And it has given me the ETA. So even on my 48 GB of VRAM GPU, it is telling me that the ETA is around 20 minutes. Wow. So let's wait for it to finish. Okay, so as was advertised, it took around 20 to 25 minutes and it's all computed. Let's try to plot it. and I'm just installing Matplotlib and using the usual code to get it plotted. So it is installing Matplotlib, and then it is going to plot it. So let's wait for it. There you go. So you see, these are all the models which we have given, and then it has generated a similarity matrix on x and y axis and how good it looks around. And the color coding tells you which is similar and which one is not similar on these axes. So all in all, really amazing model. There are a lot of other things too which you can do with it. For instance, by using this code from their repo, you can generate a phylogenetic tree plot. And so I just ran it. It just created all of these colors. It has this function to prepare the trees, and then a simple Python code, and then you can draw it by using this library. Let me go up. Sorry. So these are all the models which we have kept before. I'm using this BioPython from their repo, and then they have defined these options, calculator indexes, distribution metrics from their paper, and then you can use this philo.draw to draw this awesome sort of branch length and phylogenetic tree where different models are related to each other in a very easy to understand x and y axis graph, where you can see that there is a taxa and then there is a branch length. So this is what this amazing research is. And one of the good things which I really like about this research is that not only it gives us a paper, but also along with that it has given us the proper code to see what they are preaching actually works, which is awesome. So that's it, guys. I hope that you liked it. As I said, I will drop the link to this notebook in video's description. Play around with it. Let me know what do you think. If you like the content, please consider subscribing to the channel. If you're already subscribed, please share it among your network as it helps a lot. Thanks for watching.